So, and then we know that the important process, how DNA makes the DNA, the replication, okay, making its own copies, right? That is basically discussed in this chapter, okay? And genetics, we already know that it's the study of the genes, okay, study of variations, inheritance, how traits are transmitted from one generation to the other generation, right? And then in 1940s, it became very clear that DNA is a hereditary information. Okay, it's the hereditary information. It's the basically the important part of a cell nucleus, isn't it? So it was Watson and Crick who gave us the structure of the DNA, right? And in 1940s we came to know what is the structure of the DNA, how does the replication of DNA takes place, right? And various things about the gene control. Okay, so as I said that this is a review class, so we will not discussing the things in detail. We can discuss the main concepts, right? Otherwise, if you want to go back to each and every detail, right, you can always go back to the recording for this chapter, which is there in the Moodle. Okay, so we know that we have two types of nucleic acids. We have the DNA and we have the RNA, right? If we quickly discuss the differences between, you should be very thorough with the differences between the two. You should be knowing what is the difference between DNA and the RNA. DNA, deoxyribonucleic acid. RNA, ribonucleic acid. DNA is double-stranded. RNA, single-stranded. DNA present in the chromosomes, like along with the histone proteins, forming the chromatin. While as RNA, can we find it in the chromosomes? No ways. It's not there in the chromosomes, right? So DNA has bases like adenine, thymine, guanine, cytosine. But RNA has bases like adenine, guanine, cytosine, uracil, right? Sugars are different in the DNA and the RNA. Okay, in the DNA we have the deoxyribose sugar. Okay, and in the RNA the sugar is the ribose sugar. Okay, so difference lies in the carbon number 2. We'll go back to the structure in a while. Okay, where at carbon number 2 only H is attached in the DNA, while as OH is attached in the RNA. So you should be knowing this difference as well. Okay, so DNA is responsible for what? Storing the information, transmitting the information to the mRNA. Okay, while as RNA codes for what? It codes for the amino acids. It can act as a messenger RNA between DNA and the ribosomes. Okay, so and the base pairing in DNA, guanine with cytosine. Let me use my pen here. Base pairing in DNA, guanine with cytosine, okay, and adenine with thymine, the Chargaff's rule, right? So what did Chargaff say? The base pairing rule. What did he say? He said that the amount of adenine is equal to the amount of thymine. The amount of guanine is equal to the amount of cytosine. That's what he said, that, isn't it? So that was, as I said, that you should be knowing the difference between DNA and the RNA. This is something which is very, very basic, right? Which is expected out of you, okay? Which you should be knowing about it. Okay. So DNA carries out two important functions, of course, the replication of the DNA, which is a very important function, and then the protein synthesis. Without, as I said, that DNA carries the genetic information, right? Which is a very, very critical information for the cell, right? So without DNA, replication is not possible, right? So without DNA, protein synthesis is not possible. We cannot have proteins unless and until we have DNA. So that is why it's called as the blueprint of our life, the DNA. Okay, and every cell in our body, right, it contains the DNA, and every organism in this world contains DNA except few viruses like RNA based viruses. Is RNA present in the chromosome? We just discussed that. No, we don't have RNA in the chromosome. We just have DNA along with the histone proteins present in the chromosomes. No RNA in the chromosomes, right? Then I was just trying to tell you that you should know the difference between the sugar of the DNA and the RNA. Look at this. I was talking about this, right? Position number 2, we just have H, and position number 2, we have OH. So if the structure is given, you should be able to identify the difference between the two. which sugar is a ribose sugar and which sugar is a deoxyribose sugar. Is it clear, everybody, till now? Yeah, so you should be knowing the difference between the two. Okay, that's very, very important. Okay, and you know the sugars are basically pentose sugar, whether it's the DNA or the mRNA, both are the pentose sugar, the furanose forms of the sugars, right? So and if you remember, we discussed about how do we how do we if we have to do the nomenclature of the sugar, how do we do the nomenclature, if the sugar in the nucleotide, does anyone remember about that? If I have to say this is carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, how do I write that when it comes to sugar in the DNA or the RNA? Does anyone remember that? The carbon atoms of the pentose sugars, how are they denoted? Uh-huh. Dashes, Shata, absolutely. Very good. Absolutely. We call them prime numbers. Yes, absolutely. If you remember that, it's very, very important to avoid the confusion between, because we're going to have the bases also here, right? So bases will also have carbon 1 or nitrogen 1, 2, 3, 4. To avoid the confusion, right? So we always address them as, C1 dash as as a prime numbers, right? So dashes, so dashes will be used for this. So dashes will be used, dashes, okay, will be used for this. Okay, just to avoid the confusion with the carbon atoms present in the purines or the pyrimidines. Did you all understand what I'm trying to say? Yes, to minimize the confusion. So because if you have to, for example, if you have to name these bases, right? So you can't say 1, 2, 3 dash and 4 dash. No, you can simply say carbon 1, or maybe carbon 2 is here, not maybe carbon 2 is here and carbon 3 is here, right? So if C1 is, you have to understand that C1 means she is referring to the base, right? If I say C1 dash, that means I'm referring to the sugar molecule. Okay, this is just to differentiate between the carbons present in the sugar and the carbons present in the nitrogenous bases. Okay, we said that we have two types of bases, the pyrimidines and the purines. Pyrimidines, you should be able to identify the structures are given, right? If the structures like this are given to you, you should be able to identify this. Okay, if for example you might be given whether this is a purine or a pyrimidine, right? You should be knowing that. Okay, if you see a double ring, okay, now what is it? It's a purine. Okay, if you see a single ring, what is it? It's a pyrimidine. That's the easiest way to identify. And you should know which ones are pyrimidines and which ones are purines. Pyrimidines are what? Cytosine, thymine, and uracil. These three are pyrimidines. And only these two are the purines. You should be able to know this as well. Okay. And which one is present only in the DNA and not in the RNA? It is the thymine. Thymine is only in the DNA. You should be knowing about all these things. Thymine is only in DNA and not in RNA. In RNA, we have the uracil. Okay. So if you look at this, last time I told you, you should be able to know if I give you these two structures, this is thymine and this is uracil, especially these two are of, you can memorize these two. Because if you see the difference between the two, right? If I ask you what is the difference between the two, you should be able to know that thymine and uracil, both of them are pyrimidines, no doubt about them. But the difference is basically it lies in this methyl group, isn't it? This methyl group at position number 5, right? We have a methyl group at position number 5 in thymine, right? And there is no methyl group in the uracil. An important point to note down here. Yeah, the methyl group is a difference here, absolutely, right? Then how bases are connected to each other? We have the specific base pairing. Okay, as I said that we have guanine base pairing with the cytosine, adenine with the thymine. Between guanine and cytosine, we have 3 hydrogen bonds, right? Between adenine and thymine, we have only 2 hydrogen bonds, right? So even though they're hydrogen bonds, they're very weak bonds, right? But plenty of them in the DNA, stabilizes the structure of the DNA along with the hydrophobic interactions in the DNA, yeah, which we'll be talking about in a while. Okay. And then you should be able to know how, yeah, this structure is very important. You should be able to know what is the bond between a base and a sugar. Okay. So if you remember, we said that what is a nucleoside? Nucleoside is when you have only sugar and base together. Okay, they're called as nucleosides. Okay, any sugar and any base together, we call it a nucleoside. Okay, and how are these two connected with each other? Okay, base is always connected to this anomeric carbon. Now, you all know what is an anomeric carbon, right? And what is the bond? Very important. Bonds should be, you should be very clear with the bonds, right? It's just the naming. It's a beta N glycosidic bond which connects N1, okay, and one dash from what? From the sugar. If the base is what? If the base is what? If it's pyrimidine or purine? If it's a pyrimidine. So pyrimidine are always connected with their N1 to the anomeric carbon. An important point to note down here. Can anyone tell me about the purines? How are purines connected? Which nitrogen connects the purines? N9 chatha, absolutely. This is an important point to note down here. N1 of purine, pyrimidine connects with the C1 dash of the sugar, but it is the N9 with the purine. Okay, so that you have to remember. So you should know the difference between a nucleoside and a nucleotide, right? So here we have only base and the sugar, so it's a nucleoside. Okay, but then if we have three things together, if we have a base, if we have a sugar, and if we have a phosphate, here we have three phosphates, but in nucleotides of the DNA we just have one phosphate, right? So sugar, phosphate, base makes the nucleotide. Only sugar and the base makes the nucleoside. Excuse me. Okay. And sugar and bases are connected by the glycosidic bond, as I told you that, right? And sugars, I told you, it's a five-membered sugar, right? The furanose sugar, the pentose sugar. And which sugar it is? Is it a D or DL isomer? The sugars here. It's the, what type of sugar is this? It's the D sugar or, excuse me, D isomer or DL isomer? Remember, sugars are always D isomers. Absolutely. Yes, Sashi, very good. Sugars are always D isomers, right? So it's a D isomer, right? And then you should know the structure of the ATP as well. Very important, you should know what is an ATP. ATP, we know that it is also a nucleotide derivative. Yes, because if you see here, it also contains what? It also contains the base, it contains the sugar, and it contains the phosphate, right? So it's also a nucleotide, okay? But it's a triphosphate of the adenosine. If you see, as you can see here, we have one, two, three phosphates over here, right? So we know that ATP is an energy currency of the cell. We saw that in bioenergetics chapter, how important the ATP is, right? Now, if I ask you a question, is ATP a DNA nucleotide or an RNA nucleotide? What do you think? Is it a ribonucleotide or deoxynucleotide? What do you think? Absolutely no. Yes, it is. It's not a DNA one. It is the, yes, it is the RNA nucleotide. How do I know about that? I know that I saw the sugar over here, right? I saw this OH over here, right? So the sugar here is not, yeah, it's the ribose sugar. It's not the deoxyribose sugar, right? You should be knowing about all these things as well, right? So we know that ATP provides energy to drive many processes in the cell. We know that whatever happens in the body, whether it's a muscle contraction or nerve impulse movement or anything, right? Or you running, moving, right? So everywhere the ATP is required. Okay. Now, as I said that, you should be able to know that we have a base here, we have a sugar here, and the sugar base is the adenine here, of course. It's ATP, adenosine triphosphate. So adenine is the base here. Okay, sugar, as I said, it's a ribose sugar over here, right? And then the phosphates, most important point. No, you don't have to know the structure. No, you, if the structure is given to you, you should be able to identify, is it an ATP or is it an ADP or is it an AMP, right? And you should know the bonds. Bonds are very important. You should know the bonds, right? Because bonds are everything, isn't it? Because ATP has a capability of storing the energy, okay? And this energy is stored in this what? In this phosphoanhydride bonds. Okay, you should be able to know the number of the phosphoanhydride bonds. If I ask you a question that how many phosphoanhydride bonds are present in the ATP, you should be able to know about it. How many bonds are there? Two, isn't it? As you can see here, ATP has two, right? Absolutely, absolutely, right? You should be able to know that. Absolutely, right? So we have an ester bond over here, the phosphoester bond over here, which is first of all, your carbon number 5, okay, of the sugar is connected with the phosphate. Okay, there's an ester bond here. And other than that, we have the two important phosphoanhydride bonds, right? So when these bonds are broken, right? So they're high energy phosphate bonds. So when these bonds are broken, then only the energy is released. Okay, so ATP has the three phosphates, ADP has two phosphates, and AMP has only one phosphate, right? And once, if we have ATP, if it's broken and it will form the AMP, let's say ATP forms the AMP. Okay, so how many phosphorylation reactions should we have to recharge AMP to the ATP? How many phosphates do we need to add to recharge my AMP, adenosine monophosphate, to the ATP, adenosine triphosphate? Of course, right? Very good. So we might need two phosphorylation reactions to recharge my AMP so that it can form the ATP. Very good. Absolutely. Right? So this is trying to show you about the structure of this is the same thing what I explained. Number on the sugars are denoted with the primes, the dashes. Okay, so this is the same thing. So if you see here, I have dashes over here and there'll be no dashes in the bases. Okay, so the nucleic acids, DNA and the RNA, are the polymers, right? The repetitive units, okay, of the nucleoside monophosphates. We have only one phosphate in the nucleotides present in the DNA, or the ribonucleotides in the RNA. Not like ATP having three phosphates. No. Okay, they're monophosphates. Is it clear? So this is a typical structure of the basically a base with a glycosidic bond shown over here, and ester bond shown over here between the phosphate and the carbon number 5, and then this is the ribose sugar, a typical structure of a nucleotide. Okay. Now this is the same thing, nucleotide and nucleoside difference. One is sugar and base, and nucleotide is sugar, phosphate, and base. Okay. What is the monomer of the nucleic acid? The monomer of the nucleic acid is the nucleotide, isn't it? Sugar, phosphate, and the base. Polymer of the nucleotide is the polynucleotide, like many nucleotides together. Sugar, phosphate, base, sugar, phosphate, base together. So it makes a polynucleotide, right? Then the structure of the DNA. So what is the primary structure of the DNA? When I say primary structure, what does it mean? It means that how is how are your nucleotides arranged? Okay, so the sequence of nucleotides, okay, uh, which can begin with what, with the 5 dash end, and okay, and and in the 3 dash end, right? So this is basically the primary structure of the DNA, right? So the way we have, we read the proteins from C terminal to N terminal. Okay, for the DNA we have 5 dash 5 dash N to the 3 dash N. That's how we write it. Okay, so look at this. This is the 5 dash end and this is the 3 dash end. Okay, So can anyone explain me about the two ends of the DNA? Anybody? What do we have at the 5' end? And what do we have at the 3' end? Anybody? What does this two ends of the DNA mean? If you remember, I explained it in the previous class. At the 3' end, what is it attached to? It means no, no. I mean, if you see the 3' end, when I say the 3' end, what does it mean? Why do I say 3' end? What does it mean? What does it refer to? What does it show it to me? What does it mean actually? If you look at this DNA, this is a polynucleotide strand of the DNA. If you see here, the 3' end. Okay, always remember this. Okay, the 3' end is basically a 3-carbon end of the pentose sugar, as you can see here, which is attached to an OH group. If you remember, when we were discussing about the replication, didn't we say that the new nucleotides are added at what end? 3-OH end. Remember, in replication we said that. So your nucleotides will be added over here. Correct? Can they be added over here? No ways. You can add them over here at the 3-OH end. Okay. So every DNA strand will have two ends. One is the 5' end, another is the 3' end. So when I say 3' end, so it should come in your mind that, oh, 3' end means the third carbon of the sugar, which has an hydroxyl group attached to it. Okay. And what does it mean to say when I say 5' end? Okay. So 5' end is always attached to the phosphate group. As you can see over here, it's attached to the phosphate group. Okay, so that's why every stand has always what? A 5' end and a 3' prime end. So, and we can say that this DNA is polar. So this is a polarity of the DNA. Did you all understand? What is the meaning of 5' end and 3' end, the two ends of the DNA? Because DNA has a directionality. Okay, because if you remember, once we will discuss in the replication, DNA polymerase will read the DNA strand only in 3' 5' direction, but when it has to form the DNA strand, it will be formed always in 5 to 3' direction. So DNA strand is always read or always formed in 5 to 3', but the DNA polymerase will read it in 3 to 5', which we will discuss later on. I will not confuse you here. I just wanted to let you know what does it mean to say 5' end and a 3' end. Okay, is it clear to everyone? And then of course the bonds, the phosphodiester bonds over here, isn't it? The bonds over here. Okay, and then the we were discussing the primary structure. We can divide the primary structure into two parts: the backbone and the bases. Backbone is what? Your phosphate and the sugar. Okay, and as you see, bases are towards the center. So bases are facing each other. Guanine is here, cytosine must be here, cytosine must be here, cytosine must be here, adenine must be here, cytosine must be here, thymine must be here, right? So they're facing each other, they're towards inside, while as the phosphate and the sugar backbone, they are towards the outer side. Okay, so bases are hydrophobic, so they are towards, they want to stay away from water hating, that's why they are towards the inner side. And this part is water loving, that's why it's from the outer side. Okay, is it clear to everyone? Did you all understand what I'm trying to say? So which bond which bond links the two sugar molecules of the nucleic acid in the backbone? Which bond links the two sugar molecules in the backbone? Everyone who will answer this question, name the bond which links the two sugar molecules of a nucleic acid in the backbone. It's clear in the diagram as well. It is the phosphodiester bond, right? Which links what? What is the function of phosphodiester? It's the bond which is present between the phosphate group, this phosphate group, right, and the sugar molecule. So phosphate group and sugar molecule is a phosphodiester bond. Okay, so is it clear? So you can say that this phosphodiester bond, okay, it connects the 5 dash carbon here, the 5 dash carbon, okay, of one nucleotide. This is my one nucleotide, okay, along with this phosphate, okay, to the 3 dash carbon, to the 3 dash carbon of another. So this is my another nucleotide, right? So this is the bond here. Okay, this we already discussed the structure between DNA and the RNA. We discussed about that. We discussed this as well, the phosphodiester bond. We discussed as well the hydrogen bonds between guanine and cytosine are 3, and 2 between adenine and thymine. Okay. So the primary structure of DNA is the order of the nucleotides in the chain. Then the Chargaff's rule, we already spoke about the Chargaff's rule, right? We already spoke about it a while ago. So what is the secondary structure of DNA? The secondary structure of DNA is the ordered arrangement of the nucleic acids and the double helical strand, the double helical strand of the DNA. Okay, it forms your one strand is from 5 to 3, another one is from 3 to strand. The two strands run anti-parallel to each other. Correct? Look at this. So this was basically, the Watson and the Crick, they gave us the model of the DNA, okay, wherein they said that it's a double helical strand, okay, and there are two chains of nucleotides which run anti-parallel to each other. That means they run in the opposite direction, okay, wherein they have the hydrogen bonds between the bases connected. And each strand is made up of many nucleotides, a polynucleotide chain, having a nucleotide base and the sugar, right? And then it has a major groove, larger space, and the minor space, the minor groove. So all these things were discussed or said by the Watson and Crick, when they gave the model of the DNA, when they explained the model of the DNA. And I already explained you that the sugar phosphate back is on the outer side because this side faces the water. So they are water loving, right? While its bases are water hating. So that's why they are from the inner side. I just explained that in a while ago. So this thing also explained between, we have 3 bonds between guanine and cytosine and 2 between adenine and thymine, right? And the superstructure of the chromosome. So what is the superstructure of the chromosome? What is the main thing which we have to remember here about the superstructure of the chromosome? What we have to know here? We have to know there that DNA is what? Yes, ladies, boys, DNA is, what is it attached to? It's attached to special proteins called as the histone proteins, right? So together, DNA and the histone proteins together, it makes your chromatin, isn't it? Yes. And then you have to know that when this chromatin is available, when the cell is not dividing, at that time we can see the chromatin. You cannot see the chromatin when the cell is dividing. At that time you can see the chromosome, a condensed structure, right? And when we say, the chromatin, right, we say that 60% of the DNA and 60% of the histone proteins and 40% of DNA together makes up the chromatin. Okay, and why they're attached to each other? Because if you remember, I told you that histone proteins are very rich in amino acids. So you have to know this point as well. Histone proteins are very rich in the amino acids lysine and the arginine, which these two amino acids have the side chains which have a lot of positive charge. Okay, and DNA has a lot of negative charge, right, because of the phosphate present in it. Okay, so obviously, unlike charges attract each other. That is why histones and DNA, that's why histones and DNA, they're coiled around each other. Just because histones have the amino acids where the side chain is positively charged. As I said that, the lysine and the arginine, which are basic proteins. Very important point to note. And DNA is negative charge. So you'll have negative charge here, okay, and positive charge here. Obviously, will they leave each other? Right? They don't leave each other. They're going to hug each other together because they're of the opposite charge, right? So then it forms what is the nucleosome, right? Whenever we talk about chromosome, we know that chromosome is very long structure, and to accommodate such a long structure in a small space, okay, the DNA is arranged into what? These nucleosomes, right? And these, what is the composition of the nucleosome? Nucleosomes contain the DNA, as you can see over here, wrapped around the what octamer of the core histone proteins. Octamer, that means eight. What are those eight proteins? H2A, H2B, right? H3 and H4, right? And then H1 is here, which forms the, which is not in the octamer, which is like a separate thing. Octamer has only these four histone proteins. Okay, it forms the nucleosome. Is it clear? Okay, so this is one nucleosome bead. If you have many nucleosome beads together, look at this, many nucleosome beads together. So overall, this will be called as a chromatin, isn't it? It will be called as a chromatin. All the nucleosomes stacked together form a structure called as the chromatin, right? Is it clear to everyone? Yes, very important point to note down here. Okay. So what does the chromatin contain? Chromatin contain a DNA wrapped around the octamer of the histone proteins. Second point, chromatin is only found when the cell is not dividing, especially in the interphase, right? While as when you have to see a full-fledged chromosome, right, you can see the chromosome only when the same chromatin is shortened, it's condensed, then only you can see the chromosome. I think if you will remember, okay, that's okay. If you can memorize the hierarchy, but most important, the organization is important here. Whenever we talk about the superstructure of DNA, you should be knowing that, okay, double helix DNA, what does it form? A double helix DNA, as it's a short segment of DNA, right? So wherein you have the nucleosomes, right? So nucleosome, there are many nucleosomes over there which are stacked together forming the chromatin, right? So no, replication fork, we're not talking about the replication or a transcription yet, Mohammed. We are just discussing about the basics of the DNA, right? So the organization is important, right? So double helical structure, okay, when it's wound around the octamer of the histone proteins, it's wrapped around it. What does it form? It forms a nucleosome, right? The 11 nanometer nucleosome. So it's the first order packaging. So what do these nucleosomes form? When the many nucleosomes are stacked together, look at this. What does it form? It forms an 11 nanometer structure, which is called as a what? Chromatin. Look at this. This is a chromatin. This whole thing is a chromatin, right? So what happens? So this chromatin then forms the solenoid, wherein you have 1, 2, 3, 4, 5, 6 chromosomes in one turn. Okay. So 1, 2, 1, 2, 3, 4, 5, 6, 6 chromosomes per turn, right? It results in the formation of a solenoid. Remember this. Okay. So the idea of why do we have a solenoid structure? The same thing, solenoid or nucleosome. The idea is so that the nucleosome can fold up, right, and occupy less space. It can fit, into the nucleus. That's very important. It helps, whether it's a nucleosome or a solenoid, the idea is basically to pack the DNA in the nucleus so that it's small enough and it can easily fit in the nucleus. That is the idea of having solenoid or a nucleosome, isn't it? But remember the point here, the 6 nucleosomes per turn. Okay. So you should know the organization. So we started with the short segment of DNA, which is the double helical. Then we had a nucleosome, right? One bead of it, a nucleosome with the histone protein. Then many nucleosomes together formed the chromatin, right? Which resulted in the formation of a solenoid. And solenoid will result in the formation of the loops. It's all condensing. Okay, it's all further condensed, okay, and forms the loops. Okay, so after loops, what do we have? Does anyone remember? Loops are arranged into various bands. Remember this? These are the bands, mini band. Okay, so which is again a condensed structure of the chromatid. Okay, and then these bands finally, okay, these bands form the structure of the chromosome, which is the highly condensed form of a chromatin, this metaphase chromosome. Is it clear? So remember the organization. That okay, yeah. And this is the same thing, the organization. And finally, we have the metaphase chromosome. We start with the double strand of the DNA. Okay, then the nucleosomes, then the chromatin, then the what? Then the solenoid. Then after solenoid, we have the loops, then the bands, and then finally the chromosome. This is the same thing, difference between DNA and the RNA. So what stabilizes a double-stranded helical molecule? The hydrophobic interactions as well as the hydrogen bonds. Which bond is stronger? It's the guanine cytosine, because we have 3 bonds over there. Chargaff's rule, the concentration of A plus G equals to the pyrimidine T plus C. Right? So then this is basically the overview of the information transfer. We have the DNA forming its own copy called as a replication. But when the same DNA forms the mRNA, we call it a transcription. When this mRNA forms a protein, we call it a translation. Okay. The different types of RNAs. Do you want me to discuss this, or you already know about it? Should I discuss the RNAs? This is like a very easy thing. We know what is the function of tRNA, ribosomal RNA, mRNA, right? So mRNA is basically, for example, the coded, it carries the what? The coded information to the cytoplasm. That's the main job. Yeah, we can skip this, absolutely. You know all this. This is all easy. You know about all this. Yeah, the new RNA. Yes, Ran. I will explain that, right? Yes, no problem. Right? So transfer RNA, what is their main job? Their main job is to ferry the amino acids to the ribosome, to send the amino acids to the ribosomes. Okay. So we also know the structure of the ribosomes, right? We discussed that. So you can study the larger and the smaller subunit and their composition. So this diagram will make it easy about the larger and the smaller subunit, both in the prokaryotes as well as in the eukaryotes. Okay. So leave this. Okay. Yes. So what are your SNPs? Okay. These are like the recently, identified, or you can say recently, developed, or you can say recently found, discovered RNAs other than your basic mRNA, tRNA, right? We have these SNPs, okay, also called as the small nuclear RNA, right? So small nuclear RNA basically, it combines with the protein to form the SNPs. So you can say the small nuclear RNA, okay, plus the proteins together, okay, form something called as the small nuclear ribonucleoprotein particles, okay, which you often pronounce as the SNPs, okay. So what is the function of the SNPs? They help with the splicing, okay. What is the splicing? What is splicing? If you remember, we discussed about we need to remove the introns from the pre-mRNA. Does anyone remember that? Once you form the mRNA, okay, This mRNA can be rich in various introns, and we don't want these. Sorry, it can be rich in what, introns or exons, ladies? Introns, interrupted sequences, right? So what do we need to remove, introns or the exons? We need to remove the introns, absolutely right. So as we remove the introns, so we need to join the exons back. The job of this spliceosome, the job of the SNP is simply remove the introns from this pre-mRNA. Okay, and this technique of removing the introns from the pre-mRNA is called as a splicing. You know, the way we say cutting or splicing. So splicing is removing your introns from the pre-mRNA. And then finally, the exons are ligated together, you can say joined together to form a mature mRNA, which is ready to go to the cytoplasm. Okay, and then you need to know what are, many spliceosomes are ribozymes. Do you remember what are ribozymes? What are ribozymes? This question was there in the proteins chapter as well. Ribozymes are the RNA based enzymes, isn't it? Usually enzymes are protein in nature, but ribozymes are those enzymes where the, not the protein, yes, so the RNA enzymes, absolutely, where RNA is the main concept, where the protein is not the main important part, where the RNA is the important part, which has the catalytic activity. It's not the protein which has the catalytic activity, it is the RNA which has the catalytic activity. So they are called as the ribozymes. Okay, so in short, you can in simple words, you can say that what are ribozymes? So enzymes which are made up of RNA are called as the ribozymes, or simply the RNA enzymes. Okay, where the catalytic activity lies in the RNA. Okay, not in the protein or not in the enzyme part. Is it clear, everybody? So many spliceosomes can act as the ribozymes as well. Okay. So then we had another question about the microRNA and the small interfering RNA. Okay, so what was the difference between the two? If you remember, I told you that these are the two newly discovered what? The two newly discovered RNAs, isn't it? And what were the similarities between the two? If you remember, you don't have to know this, all this, you don't have to know all this pathway. So I just explained it during the class so that you're very much sure about you understand the whole concept, right? So you do not need to prepare this, right? So the only slide which you need to prepare is this slide, right? So what is the difference and the similarities between the two? Both microRNA and the small interfering RNA, we use them in the RNA interference. Remember the silencing, post-transcriptional gene silencing. When when you don't want your gene to express, you want to make sure that khalas, quiet, don't produce any protein, right? So both of them are part of the gene silencing. When we want to stop producing the proteins, absolutely, we call it gene silencing. Okay, or we call it RNA interference. So we use both these mRNAs in gene silencing. Okay, what are the similarities between the two? The microRNA and the small interfering RNA, siRNA, both of them are structurally similar, right? So except that the size difference is there. Okay, so this one is a little larger in size than this one. It has almost 30 bases. It has only 20 bases. Okay, rest both of them are important because both of them are used in controlling the gene expression. Okay, but then if you remember, I told you that this point is kind of very it's not the correct thing, because microRNA basically they blocks the translation by forming a, basically if you have the target sequence, okay, the mRNA, and then comes your microRNA. So we can never have mRNA as a double strand. mRNA is always single stranded, right? So when we have microRNA binding to the target sequence, automatically this gene is blocked. it cannot do translation. Okay. So so microRNA is the one which blocks the translation of the complementary target, or of the anything, for example, if we want to make this part as a, we want to silence this part of the mRNA, right? So we can use the microRNA. But your book says that it promotes the degradation. That is basically the job of this one. But you will write it, whatever is there in the book. Okay. So it's basically the small interfering RNA which degrades the target mRNA. If I have a target mRNA, I don't want this to produce the protein. So small interfering RNA can make it into what? Smaller fragments. Okay. So do you think then it will be able to make the proteins? Follow the slides. Yes, Tashil. Follow the slides, follow the textbook. Okay, but remember the information, right? You can follow the slides, whatever information. As I said, it applies to everything, whether it's a PHSF or PBio and everything. So whatever is in the textbook, even if it has not been updated yet, or if the concept is not, not the correct one, so you should always follow the textbook, right? So in short, both of them are there for regulating the gene expression, right? So silencing or blocking the mRNA so that the no translation can take place. Okay, whether it forms this double-stranded DNA, okay, complementary DNA with the target sequence, which blocks the translation, or whether it forms the fragments of the target mRNA. In both the cases, the translation is blocked. mRNA cannot form the proteins. That is the main idea here. We want to block the translation, either this way or by degrading the mRNA. Is it is it clear to everyone? This is called an RNA interference. Is it clear to everyone? Ran, is it clear? That's
Clear,
all you need
doctor.
to know
Thank
here,
you.
right? So that's all you need to know. So no need to do this pathways and all, right? Because I just explained it in a, in a lab. You know that, I believe in explaining the concept, and then for the exams, you just need to know what is there in the slide or what is there in the book. Okay. So and the application part, that these two RNAs, they have a lot of applications in our medicine nowadays, right? So they can block the expression of any viral protein, block the expression of any viral mRNA, block the expression of any RNA which is involved with the tumor formation, the cancer development. So we can silence any cancer promoting genes by using these two mRNA. So yes, application part is very, very important. Just read that from here in this slide, right? So antiviral treatment by blocking the mRNA of the virus is basically the job of these two RNAs. Okay, so that is very important, right? So we can use these two RNAs to make the anti-cancer drugs in future. Okay, using the RNAi. RNAi is basically RNA interference. These two are together involved with the RNA interference, right? Interfering with the translation of the RNAs. Okay. So then what is an exon, which is the what the coding sequence, and intron the non-coding sequences, right? We know all this. So we know all exons and introns. This is all everything is very easy, right? So moving on further, so we know what are satellites. Highly repetitive sequences are the satellites, right? We spoke about the telomeres last time at the end of the chromosome, right? Which protect the chromosome from degradation. So in short, they are what? Highly repetitive sequences, whether it's a satellite or a large satellite or a mini satellite, right? So they're non-coding sequences, okay, present in the chromosomal DNA. Okay, and we did all this. So then comes your DNA replications. What do you need to know about DNA replication? Very important part. In DNA replication, you should be very thorough with the enzymes. You should know what is the function of each and every enzyme. You should know what is a leading strand, what is a lagging strand. Okay, let me get a charger for my laptop. Give me a minute, guys. You should be able to know what is a leading strand, what is a lagging strand. You should be able to know each and every enzyme in detail. Okay. So skipping, what is a semi-conservative model of DNA? We have one old strand of DNA and then the new strand of DNA is formed. So one parent strand and one what new strand. It is called as a semi-conservative model of DNA. And that's how the DNA replication would take place. We have an old strand and then we have a new strand. Okay, so you should know lots of definitions here. What is the replication fork? What is, as I said, that in which direction is the DNA synthesized? Can anyone tell me what did I tell you last time about the directionality? Let me see if you remember. I gave you a shortcut to remember it. Read up and write down. Does anyone remember what is read up and write down, or you forgot about it? Yes, ladies. Does anyone remember that? Synthesized from 5 to 3. Okay, very good. So, but the DNA polymerase will read it from 3 to 5, isn't it? So I think we will continue this. The DNA replication is only left in this chapter, right? So we'll continue this at at 11 a.m. because I would like to discuss the issues with the 10:30 a.m. class if they have any issues with the research submission. Okay, so I'm going to stop the presentation over here. Okay, so then I will log in with the 10:30 a.m. Zoom link, right? You already have that link pasted on the Moodle, right, ladies? So we will continue with the replication in our class. Maybe at you can join back at 10:45 maybe. I think that should be okay. Okay, or if you want to join, you can, but you need to, we need to change the link now because I want the 10:30 a.m. to join first, and I need to resolve their issues if they have any research submission issues, and then we can continue with our replication. Would that be okay? Okay, so yeah, so I am going to end this class right now, and then I'm going, please join with the 10:30 a.m. link which is there in the Moodle. Okay, but you can join maybe at 10:45. By that time I will finish up discussing their issues if they have any issues. We'll continue with this one replication, and then we'll move on to the next chapter, the gene expressions as well, and finish that by inshallah by 12 p.m. today, revising it 12 p.m. today. Okay, so I'm going to stop over here.